Thank you for that intro and for joining us today for the Antibody Discovery webinar. My name is Vega Shah, and I'm the PMM for research at Benchling. I'm joined by Sanjay, who's a product manager at Benchling. Sanjay, would you like to introduce yourself real quick?
Sure. Hi, folks. I'm Sanjay. As Vega said, I'm a product manager here at Benchling. I've been at Benchling for a little over 4 years, and I've worked on a bunch of our products, including insights, lab automation, search, machine learning, and data infrastructure, and most recently studies. Previously, before Benchling, I was a healthcare and biology lead at Palantir Technologies. Vega?
Thank you, Sanjay. Like I said, my name is Vega. I am currently the product marketing manager for research at Benchling. Previously, I worked as a product manager for a lab informatics platform. And during this time, I worked with a lot of biopharma companies, doing software implementations for them and managing their data. And my educational background is in biology, more specifically in genomics. So let's talk about our agenda for today. We are going to talk about antibody drug discovery, with just a quick disclaimer that this is a pretty diverse topic and there are lots of ways to dig into it. So we'll cover some primary topics, but certainly not an exhaustive list. We'll start off with target identification and screening. We'll talk about lead selection and optimization, and then in vivo safety and efficacy. And then finally, we'll close with what Sanjay and I actually work on at Benchling, in terms of creating an R&D cloud that helps scientists manage their data. And we'll get into how Benchling helps researchers handle antibody and protein research data. And at the end, we'll wrap up with a live Q&A. All right, so let's talk about our first topic. Target identification is kind of the first step that kicks off this drug discovery process. And it's a very complex topic because there's so many different approaches to do target identification. Sanjay, you have direct experience working with this in your past role. Can you tell us a little bit about target identification and then also talk about some common pain points in doing this?
Sure. Yeah, so as folks might know already, target identification is a process by which you're trying to identify a particular target. A target meaning some biological object that seems to be causative of a disease, and the mechanism by which you would typically do target identification, at least in the distant past, is by paper review. So you would often look through biologists that are trying to understand a disease and see which, say, proteins are upregulated that are causative of a particular illness, and with the hope that you would be able to create a drug that reduces the effect of that particular protein, and thereby ameliorate the disease. That was the sort of historical mechanism by which you would do it. Today, though, there's much more modern ways of doing it, and there's a lot of pain points. It's quite common now to use retrospective analysis of patient information to try to understand from a population basis how diseases progress and why they progress the way they do. So you're often not just looking at papers, but you're also querying data from a bunch of different places, trying to integrate it together and predict which thing is causative of disease. And that causes a lot of issues. Both the qualitative understanding of a biological pathway and the quantitative understanding of how it affects a population of people.
Mm-hmm. Mm-hmm. Yeah, absolutely. And that kind of matches like my experience in the past working with biopharma companies that want to do this type of research. They're almost always dealing with a deluge of data and how to handle lots of data and collate it, right?
Mhm.
So one, I guess let's talk about the future of target identification. Where is this field going, and what are some novel techniques or mechanisms that researchers are using to tackle this sort of data deluge, right?
Yeah, definitely. So, in terms of the qualitative understanding, scientists are moving from reading individual papers to using machine learning techniques for reviewing large numbers of papers at once. That means that they can sort of like get a much broader survey of a particular disease or disease state. In addition, of course, the main goal for understanding a target is to in some sense, physically look at the target and know where you can attack it in order to reduce its effect. And that structural biology used to be something that had to be done manually. Now you can use tools like AlphaFold to speed up your analysis of the structural biology of a particular target and where you might want to attack it. So there's a lot of potential in this field. I'll give maybe one anecdote. In my past, we at my previous organization, we worked with a large pharma, and uh, our process of target identification was trying to identify gain of function missense mutations that were common for patients with a particular cancer. And that approach of uh, complex quantitative analysis was the sort of thing that we had to do one by one in the past, but can now be done in minutes today.
Yeah, that's amazing. And thank you for sharing that experience that you had in the past. That's enlightening. So let's switch to the next thing that happens after target identification, which is antibody screening. And I love that description. Throw stuff at the wall and see what sticks, because I think that's really spot on, right? You're really just doing this high throughput screening step to find a needle in a haystack. Tell us a bit about this.
Sure. Yeah. Particularly with antibody therapeutics, at least in the traditional sense, the overall structure of the protein is fairly predictable, but the specific antigen binding sites, those can vary obviously quite widely, and so the standard way that you would try to generate the ideal antibody is that you generate a large number of potential antibodies, and then you throw all of them against the target and see what would bind correctly to the target, and also not bind to other proteins. I think the not binding is also quite important because you need something that's very specific and has high affinity. The main mechanism that you would, at least in the past, generate a bunch of antibodies would be to, say, inject the antigen into an animal and produce the antibodies from there. Now there are much more modern techniques that can be used to generate large numbers of antibodies that deviate only slightly in their sequences in order to find the ideal protein.
Yeah, and so let's now talk about that perfect ideal protein, right? Which is also the step that's often called lead selection and then optimization, which is, you find that perfect protein. It has high affinity and specificity for your target molecule. Um, Tell us a bit about this process, and then also tell us what are some common issues that organizations run into when doing this.
Sure. Yeah. I think one of the things that you're trying to do in this process, as you said, is you're trying to provide or produce the perfect candidate, and that means both from a scientific perspective, like is it actually binding correctly to the target, but also from just sort of the practical. Well, I need an antibody that has certain solubility properties and certain PK properties, and so you're sort of trying to get to the optimal thing. And what will often happen is at this stage, various candidate antibodies, say antibody A and antibody B and antibody C, you only have a few of them now, are handed off to different scientists in the lab to do in vitro assessment of these various properties, whether that's affinity, or solubility or whatever it is. And that means that scientist A is going to have some data on their laptop, and scientist B is going to have some data on their laptop. And so you now have this problem of needing to, in order to actually understand what the best antibody is, combine the data together and then draw conclusions from it. So you start to see the siloed data problem, the problem of needing to track the lineage of a given antibody from its, say, earlier sequence, and not having a source of truth across disparate systems.
Mm-hmm. Mm-hmm. And I guess moving on to where we're seeing this field going. Tell us a bit about what's happening in lab. How are people solving these problems, and what future advancements are we seeing?
Sure. Yeah. A lot of the siloed data and lineage and source of truth problems are being solved with software, usually web-based software that allows you to sort of um, collaborate with your colleagues more directly and to prevent you from capturing data in a different way than than someone else. As far as future advancements, some of the things that we are starting to see include far higher throughput. And the higher throughput actually means sort of fewer scientists per unit experiment, but many, many more experiments. And so you now, it's not uncommon to see things like liquid handlers and automation systems that allow you to run screens and lead selection and optimization across thousands of wells at one time. There's also some improvements in our understanding of structural biology and our ability to use more complex instruments to understand how the antibody is performing.
Mhm, mhm. And I mean, to your point about higher throughput, especially using automation and robotics, it used to be that, that was those types of instruments and technology was fairly expensive and, required um, a lot of funding to actually set up. So a lot of the bigger companies were doing it. But now, as we see that technology advance more and more, become more ubiquitous, what I'm seeing is that it's more affordable, more accessible to have lab robots and have software that allows for automation. So it's not just the bigger pharmaceutical companies that are working on this, but also startups and kind of mid-sized therapeutic companies picking up automation in lab. Sanjay, does that kind of match what
Absolutely.
you're also seeing?
Yeah, absolutely. It's the more sophisticated techniques as we have described them is no longer relegated to the large pharma organizations. In fact, it's now considered to be quite common to do high throughput screens, even if you're a startup biotech.
Hmm. Absolutely. Okay, so we've kind of reached that point where we find this candidate, but there is a step before going into clinical research that kind of de-risks the process, and that's when we test it out outside the petri dish, right?
Mhm.
I think as biologists we understand that what happens in a petri dish and with a cell isn't exactly the same as how something functions in an entire organism. So what we need to do is really test it in an animal. Tell us a bit about that.
Sure, absolutely. Yeah, as you said, the antibody may perform or may bind to the target in vitro, but it's unclear whether it's going to have the same performance in a living species. One common example of an issue that you might see is that the antibody at therapeutic dosages does bind to the target, but causes acute liver toxicity. And unfortunately, for a drug to be useful, it needs to be able to be put in humans, most of whom have livers. And so you have to exclude drugs based on or efficacy reasons because they may perform slightly differently in the actual live animal. And the way that you typically test that is by using mice or other smaller mammals that replicate the human system, or at least replicated enough that we have some sense or some confidence about the safety or efficacy beforehand. Here's an example on the far right. This is Avastin. It's bevacizumab. It's an antibody therapeutic that was developed by Genentech Roche. And here we can see that the experiment of measuring Avastin is a drug that's used for solid tumor cancers, among other things. In this case, we have immunocompromised mice with a solid tumor cancer injected subdermally, and we measure how the tumor volume grows over time on a control substance versus bevacizumab, which is the antibody therapeutic. And you can see here that on the antibody therapeutic, the tumors grow a lot slower. And in fact, when the therapeutic is combined with paclitaxel, the chemotherapeutic small molecule drug, the tumor volume sort of flattens. It stops growing. So this is the sort of experiment that one might do. The goal of this experiment is, of course, to measure the efficacy of the drug. But probably before this study was done, another study was done to ensure that the animals actually survived at therapeutic dosages. That is to say, there was not liver or kidney or any other sequelae that were sufficiently damaging to discontinue the drug.
Thanks for that example, actually. So my background isn't quite in the animal study side of this. I'm more on the sequencing side. So I really like how you explained it with this example. It really helps me understand the importance of preclinical research. Let's talk more about that. So what what are some common issues with doing, in vivo studies? And also tell us a bit about, where do you see this field going in the future?
Sure. Yeah. I think for in vivo, it's quite interesting. Most folks who work in the vivarium, they tend to be trained specifically for that work. They're often not specialists in the drug development process itself. That is to say, they don't actually do the in vitro work before the animal study. But they are specialists in animal welfare and ensuring that the animals are dosed correctly and sample capture and so on and so forth. So there's often a internal handoff from the person who developed the candidate to the person who's testing the candidate in the animal, and that handoff means that there's likely to be a lot of complexity that you want the in vivo study execution person to understand enough about the drug, and that you expect samples from the animals to be usable by folks downstream for, say, bioanalytical use cases. So there's a lot of handoffs there, and that means that you want to be able to share data back and forth. You want to be able to share context back and forth, and allow people to make decisions across both systems if they use different systems for each. The other piece where this gets even more complicated is when you hand off your in vivo work to a CRO. This is something that we see very, very commonly in industry. There's a cost associated with setting up a vivarium in-house, and so pharma organizations, particularly smaller ones, will often work with CROs in order to actually execute a particular in vivo study.
Yeah, and I mean, I think totally validates my own experience as well, working on software implementations. Um, there's always lots of different roles that need to access data, but it also has to be sort of a combination of permissions as well, right? So it's
Mhm.
not just that everyone needs to see all the data; it's that each role needs to see the exact data that's contextual for their research. And that's especially true if you're working with someone outside your organization. You don't want to expose all your information to them. You want to only show what's necessary. Um, I've also seen that with in vivo data, it has several layers of complexity because you know it's like longitudinal. There are time series involved. There are lots of people
Mhm.
involved. All of those factors kind of come into play. So let's talk about how people are solving some of these problems. Like what are they using to manage that type of information?
Yep. Yeah, so I think there's a few places where the field seems to be progressing. The first one is that it's no longer acceptable to have sort of totally siloed systems between your in vivo work and your in vitro work. Instead, uh, uh, it's sort of the standard practice now to start to integrate the two systems together and allow you to capture data in vivo and for example, correlate liver toxicity in the animals with the specific gene sequences of the heavy chains of the antibody therapeutic. That's the sort of analysis that scientists expect to be able to do, and they can do if the data is integrated together in a nice way. The other piece is that scientists are now using more sophisticated imaging and instrumentation for their research preclinically. So things like calipers and scales, uh, even echoes, are directly tied to their computers in order to capture data instead of sort of manually having to enter things one by one.
Yeah. Great. So we've talked about the various steps in
Mhm.
antibody drug discovery. Let's switch gears a bit and talk about what Sanjay and I do at Benchling and what Benchling provides for researchers to solve some of these problems that we've talked about. So Benchling's R&D Cloud is a platform to manage R&D data. It basically, the productivity hub that you see here is your entry point into Benchling, where you can almost all teams that we work with, they pick a notebook, registry, and inventory that allows them to capture, standardize, and interlink their data. A lot of teams that we work with also use the biomolecule design app, where scientists can design DNA, oligos, and amino acids. We have a workflow app where managers can set up and manage tasks and processes. So this is where, when Sanjay was talking about handing off role from one role to another, when you're doing collaborative research, workflows can be really useful. We have an analytics app that helps uncover insights about experimental and operational performances. And finally, we have a lab automation app that allows scientists to connect physical instruments and automatically gather data straight into the Benchling Cloud platform. The other key thing, and I think we've talked a little bit about this, is the ability to integrate with other software. So we're increasingly finding that scientists want to say do statistical analysis somewhere else, but then they still need a single source of truth or a place to store that information and then share that information. Benchling is often the place where they do that because we have open APIs that allow you to connect your broader ecosystem of tools and systems into Benchling. We also have codeless configuration that allows scientists to customize Benchling to their specific needs and modalities. And then finally, Benchling's data warehouse that provides storage for very large data sets. And this is where, that high throughput data that we've been talking about that repeatedly comes up in the drug discovery world is, we allow you to store that in a place without this high cost of ownership. Now, even more specifically, related to antibody research, Benchling has worked for many years with many customers on this topic, on protein and antibody discovery. And so we've used our knowledge of doing these hundreds of implementations for organizations to sort of create this framework that we see come up very commonly, right? So what you see here is sort of a common framework we see very often. It includes discovery and screening phase with variations that represent different options that include hybridoma, B cells, phage, or yeast display, and in silico or computational screening, or any combination of these. Once that step is done, scientists move on to sequencing and preparation of the DNA. And often this is done across different teams or potentially with CROs. So that handoff is made much easier because you're on a cloud platform. The next step usually is the molecular bio group that do the cloning step, which complements the in vitro process with in silico design of plasmids. And from there, the protein teams will run protein expression and purification steps. Following this, teams will incorporate different types of in vitro testing, and that can be done, again, either in-house or at CROs, which is then followed by in vivo assays. We talked a bit about this as well just now. Testing in animals, which, again, sometimes done in-house, sometimes done with partners or CROs. So to learn more about this, please visit benchling.com. And that is my last slide. Thank you for joining us for this great discussion. And now we'll do a Q&A.